because that's really what I'm going to be talking about, is sort of our experience of seeing things in nature. And this is, I would say, something that lots of people can relate to. Can you hear me okay without the microphone? Yeah. Something a lot of people can really relate to when it comes to personal experience. To say, just when you go out into nature, just you feel, I'll use a big word, transcendence. There's something bigger than life, something that seems important and big. And so that's an experience that many people have. And actually, just as an aside, it's interesting, you know, when I was on the admissions committee for the physics department for about 10 years, and we read lots of applications. And lots of applications, including ones from overseas, would start out their essay saying, when I was a little child, I used to look up at the stars, and I would wonder at the stars, and that's why I wanted to become a physicist, which, you know, warms my heart. There's also an interesting study that says, actually they did a statistical study, that people who spend time in nature are more likely to believe in God, that they would have that, see some kind of connection with that. The question that sort of my topic for tonight is that just sort of a nice personal feeling, a completely subjective thing, or is that actually a scientific statement? Is it actually scientific to say that we see God in nature? And it's a pretty controversial thing in our culture to say that, because many people, as I put up here, would have a high wall of separation between religion and science, say that they have nothing to do with each other. And yet, we really can't make that wall so easily. First of all, because really science is nothing more than going into nature and looking very carefully. So I would argue it's not really fundamentally different in kind from what you do when you're walking in the woods. So my grandmother grew up on a farm, never went to college, but she knew every type of tree and every type of plant and every type of animal, all the different varieties and so on. And she was scientific in that way. She knew, she identified things, she observed carefully. And really, you could say science grew out of that same instinct of people simply observing carefully what's going on in nature. And so to make science something completely different from our experience in nature is putting a wall, I think, where one doesn't belong. The other side of things, which I'm not going to talk a whole lot about, is just for Christians, when we have the Bible takes place in the real world. And it says it happens in this physical world that we live in. And so you can't completely separate it from the physical world. If it says there was such and such a type of plant or there's such and such a thing happened in the world, then in principle it relates to the real world. And so as I said, the God of the Bible is the creator of nature. There's a connection. It might not be true for every religion, but for Christianity, there's an intimate connection between God, who created the world, and the nature that he created. Now, one of the things that people often would say is, well, my religious feelings when I'm walking in nature are just sort of a subjective thing that can't be quantified. It's just my feelings. But actually, there's a long history of things that I would call built-in functions that people have that at first are subjective and then become very quantitative. So, for example, we have a built-in sense of temperature. And we have a built-in sense of something being heavy. And so maybe 200 or 300 years ago, you could be sitting in a room and one person says, I think it's hot. Another person says, no, it's cold. And they're debating and says, well, it's just subjectively, that's your opinion. But now we have a Celsius scale, and we can say, no, it's exactly 72, that's how hot it is. And we can quantify that and say, that's the temperature. In the same way, somebody might say, well, this is heavy, and somebody else says, no, it's light. And, well, then you can quantify that with kilograms, and you can say, well, what used to be subjective is now quantitative. Now, at a little higher level, we also have built-in functions for face recognition and voice recognition. Little babies, from the time they're born, recognize faces without being taught. Actually, maybe even 20 years ago, somebody would have said that's completely subjective. But probably now, some of you are probably software people, right? I gave this talk recently to people there was a bunch of software engineers, and they were nodding their heads. The new iPhone is going to have face recognition so that you look at it and it says, I recognize you, and it's not somebody else, and it unlocks your phone. So face recognition is getting pretty good. And the way that it's done is by quantitative algorithms, by calculation. So it takes a bunch of data in and then does quantitative calculations and says, this is a face that matches this person, and so on. And voice recognition is actually a little bit easier than that, and is also possible. So you could ask then, do we have a sense of recognizing God in nature? Is that like one of these senses, where there's something built in that's hard to quantify, but it's maybe not impossible to quantify, and it's actually nevertheless very real? Now, some people will argue against that by saying, well, you can go wrong. People have said this is from God, and then we found out, well, that's much easier to explain, and so on. Well, that's actually true with all of our built-in functions. We can have mistakes in our built-in functions. So for instance, as I said, babies have a built-in function of recognizing faces, and it's very useful for finding your parents and recognizing your mother, and so on. On the other hand, there's been face recognition that we sometimes see in the clouds or in patterns in the wallpaper or on toast. So every now and then, probably about every 2 years, somebody sells on eBay a piece of toast with a picture of Jesus and sells it for $10,000. And there's a museum in Las Vegas that collects all these pieces of toast, so you can go there. But from looking at this, would you say there's no such thing as a face then? No, you would say that there is something that is a face. The face recognition function is working overtime trying to pick up something that isn't there. But it's not because there's no faces to be seen. It's just because of trying too hard using a built-in function that you have. And so the fact that religious people might say something a little silly about one thing in nature doesn't mean there isn't anything to be seen. It means maybe we're trying a little too hard sometimes. So here's the question. Are people wired to recognize design and beauty? Is that a built-in function that we have? Now, here's a picture of something that I assume you have no idea what that is. Okay? But you probably would be able to say that is something that was designed. Right? It wasn't formed randomly by metal exploding out of a pot or something. All right? So you have a built-in ability to recognize something made by people, something which is designed, even when you don't know what it's for or what it does. Okay? Now, we could go another step, sort of like that face recognition problem. We could say, well, here's something that has a lot less design, but we're pretty sure that we could recognize design. This is what archaeologists do. They go find a whole bunch of old things, and they try to recognize human artifacts and say, well, this one looks like it was really designed. Now, this one's a little bit more on the hairy edge. It's like looking at a face in the toast, maybe, okay, where you say, is it design? Now, archaeologists actually would argue that that is, in fact, a knife, that is a design tool. And they would have quantitative standards for how they would argue that. But some of us would look at that and say, I'm not really convinced. It doesn't really, but we have sort of a threshold of where we'd say, okay, that one for sure, but that one's kind of iffy down there. But nevertheless, there is something to be seen. Design is a thing that we can recognize when we see it, even if we can't quantitatively define it so well. So this basically gets us into the topic of what's called intelligent design. Now, you're all graduate students, most of you are graduate students, I think. You probably have heard the bad word of intelligent design, right? It's an evil movement of people who want to take over the government and turn us into Nazis, right? You've heard that? No? Okay. Well, that's this kind of thing that you hear out there. Basically, the intelligent design movement is just what I said, is saying that what we see is actually there to be seen. That the design that's actually a scientific statement to say that things that look designed actually are designed. And that's basically the bottom line. That's the foundation of the whole thing. Now, sometimes people will misunderstand the arguments that are being made. So for instance, sometimes a very simplistic version of this argument is made where people will say, well, anything that's improbable must be designed. But that's not what the intelligent design movement is saying. So you could say if I go to a shopping mall and I meet a friend that I haven't seen in 10 years, you say that's improbable, but that's not evidence for design, for some master plan that brought us together. Maybe there was, but that event is not evidence for that. And so it's not sufficient to say that anything improbable is evidence for God, and that's not what the argument is. The argument is saying, first, we see something that looks designed with this, you could say, this built-in sense. And then somebody gives a counter-argument and says, no, that happened by utterly random forces. There was no design whatsoever. And then you can say, if it was random forces, what's the probability that it would come about? Do you see how the argument goes? So you're not saying everything improbable is evidence for God. You're saying, I see specific things that I think were designed that look like they were made by the hand of God. And then if you tell me of something else, I think your something else is very improbable. Okay, so, and then we get down to do numbers and say, all right, how probable is it? Now, I'm not actually going to do any numbers tonight, but a lot of the articles you read look at numbers. Again, it's not because every improbable thing is somehow designed, but rather, if something has design, it's very unlikely for it to have appeared just by random processes. So what I want to do now is actually just take an excursion into the microscopic. We're all used to, at the macroscopic scale, of having these kind of feelings. So we go into nature and we see trees and we say that's beautiful, or we look up at the night sky or the clouds or whatever. And oftentimes the argument will go, well, you're just impressed with those things, but actually if you understood the physics, if you understood what was going on at the microscopic scale, you would understand how all of this came about very simply and randomly, and you wouldn't be so impressed. Okay? So what I want to do is actually look at some actual things that are in the cell. And the interesting thing about these types of things is that there's no lower level. Once you get to the cell, okay, you go one level below that, you're talking about atoms, okay? And the atoms now are not going to, form little machines and, and so on by themselves. So basically, at the cell level, we're at the lowest level where we're looking now at the proteins, the machines of the cell, and so on. Historically, there's been a long history in biology of people starting at the large scale and saying, well, I don't understand this, but if I understood one level lower, then I would understand it, and it would be simple. And then they go one level lower. Somebody here mentioned this, right? You go one level lower and say, oh, no, that's also amazing. But if I went one level lower, then I would understand everything. And then you go one level lower and you find it's even worse than I thought. It's more complicated, more well-designed, and so on and so forth. And so finally, we're at the bottom level. We're seeing how all the machines are working. And there's still a lot to be learned, but there's a lot fewer boxes to hide things. And so let's just go and charge right in. So I believe that I can start this magically here. There we go. Okay, so I'm not going to talk through all these machines. Just to say again that these are things which are based on calculations. So let me just talk you through a little bit of this here. I'm going to wait till the next cycle comes around. One of the things a lot of people don't know is they used to have this picture that molecules just floated like in a liquid inside the cell. That almost never happens. It's actually a very extremely compact factory. And so you see these are like little highways. And so when you have a protein that's made in one part of the cell, it's tagged with a message like Federal Express, and it's sent on a highway to its exact place where it's supposed to go. And these are the highways that are built and unbuilt now this is literally how it looks, okay? This is several people's PhD thesis just to work out this, okay? That is carrying this vesicle, which is holding inside of it a delivery that's going to go somewhere else in the cell. it's been tagged and it's going to run up this, this highway and take it where it's supposed to go. This is now a messenger RNA, which is being read out. Many of you may be very familiar with that process. You can see there now it's making a protein. We're going to come back to that one. Again, this is the RNA transcription process here. Every time little kids watch this, they say this guy is really cute. Right? And I actually know a guy in biochemistry who made puppets that he sold of the kinesin molecule. So there's a lot of processes in the cell which are triggered with signals. And a lot of times people say, well, this molecule does this or does that, but they haven't even begun to scratch the surface. How does it know when to do it? What starts the process? There's a whole level of things, of signaling to say when something should start and stop. Okay, I'm going to move on to another one here. And this one is going to focus in just on the process by which DNA is read out to make new machines. So once again, I think I can start this here. Okay, so the first is going to be the packing of DNA. So DNA molecule is about 3 feet long in each human cell, and it's got to be rolled up and compacted to be less than a micron, all right? That generates enormous pressure. This thing is stiff, and so when you coil it that way, it generates huge pressures, many times the atmospheric pressure. And so you have to have other machines to wind it up. Okay, well, when you do that, then you have to ask, how do you ever read this out? How do you ever, pull out a part and read it? Well, it's like a tape in some ways. You have to pull out a part, read it, and then put it back in again. All right, so in this process here, this is being wound up for cell division. And you can see it's rolled, and then the rolls are rolled, and so on and so forth. You can turn up the sound a little bit. Okay, now in the next part, it's going to actually show how the DNA is copied. And this is one I'm going to spend a little more time on. Okay, so let me see if I can pause this here. Oops. What's going to be going on here, there's 2 strands of DNA, as I'm sure you all know, being graduate students, and they're normally pinned together. Well, when you want to copy it, you're going to pry apart those 2 strands, and there's a little copying machine that copies each strand of DNA. That process has to have error correction in it, just like you have in computer codes. There's another machine that follows the copier that fixes the mistakes whenever it finds mistakes. Now, one of the tricky things is the DNA is a spiral. So the copying machine, if you think about it, you've got 2 spirals, okay? So the spiral for one is going to be copied moving inward, and the other one is going to be copied moving outward. Okay, so you're going to have a problem in that the one moving outward, how do you handle that when it gets to the end? All right. So the way this is done is basically, if you think about holding a bunch of rope, okay, you pull out a loop, it copies along until it reaches the beginning, and just before it hits, you pull out another loop. Okay, and so this is what's called the replicase DNA. Okay, so you see the loop being pulled out. So one of them is just copying in the same direction the whole time, but the other one is copying the other direction. See how unlinked it there and then starts a new strand. Okay, so all of this is the machinery that's doing this weaving to copy out the DNA. And again, this is not just artist conception. This is worked out by a lot of research and computation. And every one of those dots is an atom. So you can see you're feeding in a double strand here, and then you've got two double strands going out at the end. Then you break off that one, and then you start another one coming out again. Okay, so the crucial thing here is that this has to work before natural selection can work, all right? Because natural selection works by copies being made, all right? So natural selection assumes that you have something that copies and makes progeny, and then variations happen in those progeny, okay? But you have to have the copying work before that happens, all right? And not only that, but the copying has to be almost perfectly error-free. So if you think about copying any file, say, copying a JPEG file or, something like that, and you do that, 10,000 times, information gets lost in most systems. And so in regular computer systems, that's fixed by having error correction, where after you copy it, it does what's called checksums and other calculations to make sure it was copied correctly. All right, that happens. That's part of this whole system here. Okay? If that didn't exist, then the whole thing would break down within 100 generations, and you wouldn't be able to have any kind of reasonable things going on. Now, for sake of time, I'm going to skip some of these other movies, but just think about the intricacies. I'll just show this one. It's fairly quick. This is the power generator of the cell. Okay? This is basically, it can run in two directions. It can either take sugars inside of the cell and use it as a pump to pump hydrogen, or it can take a hydrogen difference and use that to then generate sugars. It's a rotary engine, basically. And without this, there's no energy source for the cell. And it just sits there and does nothing. And one of the amazing things is the exact fit of these proteins that are made somewhere completely different in the cell to the ATP, ATP to create a template for that chemical reaction to happen. And again, this all has to happen before the cell is living, because this is what gives it the energy. All right? This is basically pumping in the sugar. Okay, so I could go on and on. But these are some amazing, amazing things. And this one here was actually done by a friend of mine in New Mexico. This is the close-up of that DNA copying mechanism, all right, where this is, again, a model generated at great numerical computational effort. And you can see it's like, moving along piece by piece. So it's pulling things out of the air, or out of the background, and putting them on to copy a single strand and make it into a double strand. Now this here basically shows you the fine-tuning that goes on in this. If you look at this here, the DNA, you think about it like walking on a railroad track. If you walk on a railroad track, you have to walk at the right pace in order to hit the ties, right? I don't know if you've ever did that, right? If you don't walk at the right pace, you fall in between the ties, right? So if this little spacer here isn't exactly right, okay, then this thing gets out of sync and it doesn't put the atoms in the right place. And again, this machine, this little polymerase here, is made in a completely different part of the cell from where the DNA is residing. And so it's not like it's measuring the DNA to fit it. It's made somewhere completely different, and then it comes and it's close within with tiny fractions. Okay, so let me move on to sort of talk about more about general arguments then. So as I said, saying that we see God in nature is a scientific, so lots of people would be okay if I said I see God in nature, but that's just my subjective experience. If I say it's a scientific statement is where people get upset, because in our society, science is the adjudicator of truth, right? And so if I say I see it's a scientific statement, it's like I'm saying, no, it's really true. It's not just sort of my personal feelings. And that's what bothers people. Well, what are some of the objections? I have three, and there's probably more that maybe you might think of we can talk about in the Q&A. The first one is that it's a science stopper, that people who believe this will just stop doing science because they'll just throw up their hands and say, it's a miracle. Another one is that it doesn't make any useful predictions. It's just, sort of window dressing. And the third is that it crosses the lines of religion and science, as I talked about at the beginning. So let me talk first about the science stopper argument. And there's also a fourth one I may have time to get to. Did this argument make people not do science? Well, historically, the scientific revolution came out of the Reformation and Christianity in Northern Europe. Was that just a coincidence? Most of the scientists at the time not only saw it as not a coincidence, they saw their belief in God as actually driving their interest in nature. And so what's interesting, though, some people would say, well, of course they were all Christians because everybody was a Christian in those days. So the question is, did they see their science affecting their Christianity and vice versa? Did they see their religious faith having an effect on their science? And lots of them did. So for instance, Isaac Newton, you may not know, wrote 2 books on science and 4 books on theology. Now, his theology is actually not that good, but it shows that he was very interested in it. And so he was not at all seeing a contradiction between the two. And he offered various design arguments, and I'm not going to read all these, but he says, for instance, in the absence of any other proof, my thumb alone would prove me of God's existence. Just looking at the marvels of this, rotating, bending, and so on. And then he also, as many intelligent design people have argued, looked at the eye and said, did blind chance know that there was light and what was its refraction, and fit the eyes of all the creatures after the most curious manner to make use of it? The eye, and I'm an optics person, that's what I do in my day job, the eye is really a stunning thing, because it's not even, you could say, what good is half an eye? You could say, what good is 99% of an eye? And so I'll just give you a little, because you're graduate students, I'll give you a little physics fact here, okay? In order for the eye to be useful, you have to have the surface of the eye be smooth. You look at optics, okay, they're smooth. How smooth they have to be? Well, they have to be smooth, they have to be, have small variations compared to the wavelength of light. Okay, and the wavelength of light is 1/2 millionth of a meter. Okay, it's about 500 nanometers. Okay, so to be smooth enough to do imaging, you have to have about 50 nanometer smoothness, about 1/10 the optical wavelength in order to see anything. If that's not the case, you have what's called frosted glasses. That's what happens when people have cataracts. Okay, they have roughness in their eye. You can't see anything. Okay, if you take a piece, sometimes I bring a piece of frosted glass, but if you have a piece of what's called frosted glass, that is polished to a 1 micron roughness, which is just twice the wavelength of light, and you can't see through it. People put this on their bathroom windows and so on. It turns out that you can't get molecules to be only 50 nanometers smooth. So how is our eye that smooth? The only way you can do it is by putting liquid on it. Okay? So there's this ornate system of putting teardrops on your surface of your eye. So if your eye dries out, you can't see, all right, because you're more than 50 nanometers unsmooth. So in order to make this whole thing work, you have this extremely smooth biological surface. Then you continually drip water on it so that it's smooth to 50 nanometers. And you think about it, there's nothing else in nature that's that smooth. Okay, only eyes are that smooth, and eyes have to be that smooth or else they don't work. Otherwise, you're just saying there's bright or dark, and that's not too useful for me. And so there's incredible things that go into just being able to get an image. And we could talk about a lot of other aspects of the eye. Leibniz, the father of calculus. Some people don't consider him a hero. They didn't like calculus. He's sort of the inverse. His science wasn't that great, but his theology is actually not bad. So he actually wrote some very good theology books, made a very big impact on philosophy and theology, and also on mathematics, obviously. And again, he says, we believe in God. In reflecting on the works, we are able to discover the one who did the works. Okay? So he's not making this high wall of separation, saying, well, my personal belief is in God, but it has nothing to do with what I see in nature. He's putting the two together. William Harvey, this is a really great example, considered by many people the father of modern medicine. He actually used his design principles to make a discovery. Because he was looking in the human bodies, they were working on corpses, and he found all these one-way valves. All these, and you can probably tell a lot about this, right? These little, in your veins, you have these little flaps. And he reasoned, as he says here, providence, that's an old-fashioned word for God, would not have placed so many valves without a design. And so he assumed they had a purpose. And therefore he said, what's that purpose? What are they good for? And he reasoned from that then and found the circulation of the blood in the human body. And then people were often running in human anatomy. William Thomson, again, very famous father of thermodynamics in a lot of ways. Kelvin is named on all kinds of things. You see his name popping up all over the place. Very strong Christian man as well. This is now in the 1800s. Makes an interesting point. I won't read the quote, but he basically says a person who argues against belief in God is himself a contradiction because he himself is using a fantastically well-designed brain in order to make that argument. And so he himself is contradicting even his own premises by his existence. Now, I'll just run through these. In the 20th century, it didn't stop in the 1800s. Three very well-known Nobel Prize winners, Arthur Compton, Charles Towne, Richard Smalley, Bill Phillips, who I know personally, is a more recent Nobel Prize winner. All of them strong Christians who would say they believe in God. And again, if you notice the way that they talk, they say that God reveals himself in his creation, right? Or Richard Smalley, it is increasingly clear to modern science that the universe was exquisitely fine-tuned. That's another word for design. Wernher von Braun, for the engineers out there, father of space travel, also said that God reveals himself through creation, that he sees God in nature. Okay, so my point here in listing all these people is to say there's many people, there always has been, people who are strong Christians for whom it doesn't stop their science, and they don't see it as they're contradicting each other, but just the opposite. They're saying, I see God in what I'm doing in my science. Now, what about the argument that it doesn't make useful predictions, that it's not useful for anything? I think this really gets, and maybe in the Q&A we can talk a little bit more about this. What does it mean to do science? For a lot of people, the only thing that counts as science is a history. To say, so the example I use is if I say, I went to the store and I shopped for butter there. And what's the explanation for me doing that? Well, one level would be, well, I got in the car, and when I turned the key, explosions happened of gasoline inside the engine, which caused the wheels to turn, which then carried me to the store. Okay, that would be what we call a causal history explanation. Okay, most of us would say, well, that's not the whole story, though, right? Another explanation would be, I went to the store to get butter, right? A final cause, a purpose, all right? And a lot of times people don't want to talk about those. But actually, if you're in the biology field, in certain fields such as systems biology, you may be aware in the last 20 years there's been an enormous revolution in the biology field in embracing what is called teleological thinking, that end cause. And so if you go to many, many biology departments now, they will embrace what's called top-down thinking, where they say, what is it for, okay, and what is it good for? So here's an example of how a design assumption would affect science. Suppose that somebody gave you a chip like this, and they said, here is a chip, and I want you to figure out how it works. Okay, now there's two different assumptions you could be told about this chip. One person is given this chip, and they're told, this was generated randomly, so we just randomly cast circuits on the board until something worked, and then we kept the ones that worked. All right? Another person is told about the chip. This was made by a master designer who is like super intelligent and did everything perfectly. Now, could you see that that would lead to different approaches to understanding the chip? Okay? So if you assumed it was randomly generated and you found something you didn't understand, you'd be much more likely to say it was probably doing nothing. Whereas if you believe this is made by a master designer, every little thing you tend to assume is doing has some purpose to it. And so in fact, biology has moved toward embracing a design paradigm of saying when we look at biology, we actually will assume every little thing is doing something. And that's the reason why they assumed, they didn't used to assume that. They used to assume that things were full of vestigial, non-working junk. So when I was a kid in the 70s and 80s, we were told that the human body was full of junk. Lots of things that weren't doing anything at all, were half working, and so on. And that paradigm is dead in the biology field. Basically, everybody now views everything as highly optimized. And so I put a quote here. This is what you see in the literature. And if you're in biology, you may be familiar with this. This is great. This is a paper I just pulled out to quote here. This is a paper in which he's basically saying that biological systems have optimal function. And so he's writing a paper in order to explain the highly organized, optimized engineering principles of this. And he says, we're going to explore the important design features, but then he feels it necessary to put a parenthesis with no implications of an actual designer that he sticks in there because he wants to make sure no one mistakes him, because he sounds so much like an intelligent design person. If he didn't put that in there, he might not have been able to get the paper published because somebody would have thought, and this actually happened, was it just last year? There was a paper from China on biology where it was sent into a journal and accepted, and in the journal, they said they made the mistake of switching from design to designer. And so they were saying, well, the design of this cell is such and such, and they were on good ground. And then somewhere along the way, they slipped and they said, and the designer would have done this. And this was published, it got through. And then there was this huge outcry that there's these intelligent design people in China. And so they had to change the paper to say, no, not designer, just design, and retract that paper. Okay, I'm not going to read all these quotes here, but here's just one. Bill Bielek was a professor at Princeton who does biophysics, and he was visiting our department a few years ago, and he sounded like an apostle for the intelligent design movement, talking about how perfect everything is. And he talked about bats as just an example. Okay, so bats use sonar hearing, and some people did an experiment to see how good is the sonar, how good is the time resolution. And it's really kind of comical. You know, for me in physics, it's a funny experiment, but like they had the bat there and they threw things at it, and then, little flies at it, and then they looked to see how quick it could react, and they would do it quicker and quicker and see what its reaction time was. It turns out its reaction time is 10 nanoseconds, 10 billionths of a second, all right? Basically, Bill Bielek proved that that is as good as is physically possible because of the motion of the atoms in the air. You can't use acoustic waves to get any better resolution than that. Okay, so it's as good possible sonar as is physically possible to have at room temperature in air. And so that's just an example of, again, something sort of macroscopic. Now, in biology, as I said, many people have said, yes, we're starting to embrace this paradigm of top-down thinking to say every little thing is there for a purpose. Now, some people would say, and I would say the standard line is, well, the reason for that is because evolution is so darn good that it gets rid of every inefficiency. The problem is there's a catch-22, and this is where my own research actually comes in. So that's actually a paper. Oops, I keep doing this, going backwards here. Okay. I did some numerical research on this a few years ago, and here's the catch-22. Catch-22 is from a movie, by the way. If you don't know that reference, don't worry about it. Basically means you can't win no matter which way you go. So if natural selection is really, really good at weeding out inefficiencies, then nothing ever evolves, right? Because as soon as you get anything slightly different, that one gets killed off, right? So for instance, suppose I want to evolve an eye, and I have half an eye that's not doing anything yet. all right, then I'm just going to, by this scenario, if nature is really efficient at getting rid of inefficiency, then that creature with half an eye just dies. On the other hand, if you say, well, let's make it not too efficient at getting rid of things that don't work, so we allow things to change, now you can get evolution, okay? But now this is where numerical models come in. You can say, well, there's a balance here of how efficient it is at killing off the inefficiency versus how much you can tolerate of these things that don't work, okay? And it turns out that it's a very, very difficult problem. And essentially, in this paper, is a numerical model. And of course, numerical models can always be wrong. But in this model, we showed that you basically would expect that we should be full of vestigial things. That in order for evolution to proceed, which you can make the model proceed and evolve, but in fact, you should be like 99% vestigial. And in fact, we're very far from that. And so people who argue that nature is very efficient at getting rid of things that don't work haven't really thought through the questions about how difficult it is to do that without stopping evolution altogether. If you make it too good at killing things that don't work, nothing ever changes. You burn in exactly what you have. Actually, you could argue the opposite, that many people who believed that evolution proceeded this way expected exactly that. They expected that the body would be full of vestigial stuff. And so for many years, there was the argument that the DNA was mostly junk. And then in the last 10 or 15 years with the ENCODE project and so on, they've been now finding all that junk is doing stuff. It's regulating signals and things like that. It's not being used to copy into proteins, but it's holding other information which is used for signaling, and we don't even understand the whole signaling process. Okay, so let me finish with this then, the argument that it crosses the lines of religion and science. And this one, I just have a very quick argument. This is what's called a demarcation argument. I win by saying, well, I'm going to define what you're doing as religion, and therefore it must not be valid. It's basically the amount of this argument. So drawing lines in the sand is something that people like to do. But in fact, real scientists hate that. You know, as a physicist, you know, I always like to say, I just want to believe what's true. You know, I don't care whether you call it this or call it that. You know, so if somebody says, well, that's engineering now, you're not allowed to do that because you're a scientist, I'll say, well, I don't care. And if somebody says, well, that's philosophy, you can't do that because you're a scientist and the philosophy department's over there, I'll say, I don't care. You know, and if somebody says, well, it's religion, I'll say, I don't care also. And in fact, you only have to pick up a popular science book by Stephen Hawking or any number of people, and they'll talk about God all the way through. Physicists in particular love to talk about God. So for instance, here's a couple examples. You know, Albert Einstein, Stephen Hawking talking about the appearance of the world. And Hawking, of course, arguing against God. But he's not saying, I'm transgressing a boundary here. How dare I talk about religion? He feels free to go into the religious sphere and to say things about the religious ideas on the basis of his science. So let me just finish with some questions which we could discuss, or there are other discussion questions as well. Can you trust your design sense? Are you seeing something? In general, our bodies sense things that are there to be sensed. Is design our sense of seeing something that is designed and beautiful, as people said, is that unlike all of our other senses? Is it something that's just my personal opinion, or am I really sensing something that's there to be seen? Is it wrong to cross religion and science, this demarcation line? And lastly, if I don't believe in God, do my feelings against God cloud my judgment about science? Do I actually not want to follow the science where it leads, but actually I'm afraid I don't want to believe in that, and so I'll look at science differently. Okay, so I'll finish with that, and we can do some Q&A. Thanks.